Okay, today we will be talking about advanced genetics and what happens when a certain trait may or may not follow one of Mendel's laws. Most human genetic traits aren't going to follow that one allele dominant and recessive rule. So today we will talk about when traits maybe are not completely dominant to another, or when there's multiple alleles that will influence a certain trait, or when you do have one gene but it can have multiple effects. So that is really our focus today. Remember that dominance doesn't always mean more. I think this is something that is like a common misconception in genetics, that a dominant allele is always going to be more present in a population. And that really isn't always the case. And if you look up, I think you'll find when people present their Mendel at DHS presentations that just because something is dominant doesn't mean that we see it in, the United States population more. An example of this would be polydactylism, which is a person who's born with more than 5 fingers or toes. Really, that trait, polydactyl, it is a dominant trait. But we only see about 1 in 400 children born with this trait any given year in the United States. So it's important when we're talking about traits that we kind of forget that misconception and we just take the traits as they are. The first type of advanced genetics we're going to talk about is something called incomplete dominance. And this is something we talked about when you were freshmen, so you may remember this. This is when two traits, when they come together, they blend instead of one being dominant over the other. You see this in carnation flowers a lot. And so if you take a red carnation flower and cross it with a white carnation flower, when you get that heterozygote population, or, flowers that have a heterozygous genotype, you're going to see all pink flowers. And then when you cross those pink flowers together, or heterozygote times heterozygote, instead of seeing a 3-to-1 phenotypic ratio, you see a 1-to-2-to-1 phenotypic ratio. And so in this case, the genotype and the phenotype ratio would be the same. And so when you think incomplete dominance, just think that we're blending two colors together to make a new color. Codominance is when two alleles equally affect the same phenotype. And so this is something that's not blended. You actually, in some cases, will end up seeing both alleles expressed equally at the same time. And so blood type is really interesting because we kind of see a couple different types of dominance, or a couple different types of inheritance in play. We see dominant and recessive, we see codominance, and then we also see this multiple alleles, meaning that for this locus or for this gene, you could have a combination of 3 options. You could be capital IA, capital IB, or lowercase I. Don't ask me why they chose the letter I for blood type. I do not know, but that's what we go with. So these letters, what they represent, or what they're coding for, are the type of antigens or glycoprotein antigens present on red blood cells. So if a person has type A blood, this antigen on their red blood cell is going to be type A. If they have B, it's type B. If they have a capital IA and a capital IB as their genotype, then they have both types of antigens on their blood. And then if you have a lowercase I, you have type O. And so if you're recessive for both alleles, you have type O. So let's kind of talk about the genetics of blood type. So if you have type A blood, you have 2 different genotypes that you can have. And that's, you can be homozygous for type A, or you can be heterozygous for type A. But this I capital A is dominant over that lowercase I. So if this is the case, you are going to have type A antigens on the surface of your red blood cells. So you would have, those A glycoproteins on your red blood cells. And then you would have anti-B antibodies in your blood. And even though that we're not really talking about what's present on the red blood cell, the antibodies that are in the blood matter when it comes to donation status. So then B is going to look a lot like A, except you have the B allele instead of the A allele. And then that's going to have type B antigens on the surface of the red blood cells. And that will give anti-A antibodies in the blood. So then AB, this is where those alleles are codominant, or where they're both going to be expressed. So if you're capital IA, capital IB, you have both type A and B antigens on the surface of your red blood cell. And in this case, you have no antibodies in your blood. And this is going to be really important when it comes to blood donation status. And then finally, for type O, where you're little I, you have no antigens on the surface of your red blood cells. And you have anti-A and anti-B antibodies in your blood. And so blood type is a great example of all these different types of inheritance. It's also a really good example of the fact that dominant traits are not always seen more in the population. Actually, type O is the most common blood type we have in the United States. I think it's a little over, or right around like between 55 and 60% of people have type O blood. And so that just shows you again when a recessive trait can really be more present in a population even though it's recessive. Now, when we're talking about donation status or what type of blood can give to what type, hopefully you guys all know that type O blood is the universal donor, meaning that they can give to any of the other blood types. And the reason why they can do that is because they have no antigens on the surface of their red blood cells. So if those red blood cells are put into other types of blood, they're not going to react with the antigens from those other red blood cells. And so a B blood is the universal recipient because it can get blood from type O or A and B because it has both types of antigens on its red blood cells, and it also has no antibodies in the blood that could be cross-reactive. This is just one of my favorite pictures because I think it really shows a good difference between incomplete and codominance. And so if you have a red and a white flower, and if the product of that cross is a blending of those two colors, which makes a new color, that's incomplete dominance. If you still see both original colors in the new flower, like this, where we see streaks of white and then we see a streak of red, then that is codominance. Both of those alleles are being equally expressed, and they're not changing. Pleiotropy is when one gene can affect more than one trait. And what that means is that you have one gene, you have two alleles, but you can see more than one phenotypic characteristic. And we see this most often in achondroplasia, which is dwarfism. And so achondroplasia is inherited dominantly. And so if you have a parent who is a dwarf, donated by with this capital A, and then a normal height parent, little a, and you crossed those two individuals, you would get offspring, two that are dwarf and then two that are normal height. So 50% dwarf, 50% normal. Now, if you have two dwarf parents that are crossed and we do the same thing, we get the same thing, a 1 to 2 to 1 ratio of genotype. However, that is a lethal combination. So anytime there are two dominant alleles present, that would be a fetus that would not develop. And so instead of, having that 1 to 2 to 1 ratio or that 3 to 1 phenotypic ratio, we end up with a 67% dwarf and 33% normal out of that cross. Epistasis is interesting. This is a way that a lot of different coat colors are determined in different types of animals. But epistasis is when you have one gene that is going to mask another gene. And it's two separate genes, but one kind of talks over the other one. And so in mice, this works with two genes. We're just going to call them C and B. And so the gene C is coding for either pigment or no pigment. If an individual is homozygous recessive for little c, it does not matter what this B gene says. It is going to express no color or no pigment. And so we can see this down here in this cross where, you have B's that are being expressed there, but because we have these lowercase c's, that is going to give you white mice. So if there is a capital C present or if there is pigment present, that's when it will matter what the gene on the B says. And so it's either going to be more pigment, capital B, or less pigment, brown. And so we can see here that anytime you have a heterozygous for B or you have a homozygous dominant for B, you're going to have a black mouse. And anytime you have lowercase b's, you're going to have a brown mouse. And so a way that you might know that this was occurring is if your phenotypic ratio is a little off. And so here, if we crossed two heterozygous mice together, we would expect there to be a 9 to 3 to 3 to 1 ratio. However, in this case, we see a 9 to 3 to 4. And so that's just one example of epistasis. Labrador retrievers is another way that we see this. And so they have two genes, we'll just call them E and B. So E is expressing pigment, and then B is expressing the concentration of pigment. And so this is one where it's a little bit different because you can see differences in yellow labs based on what that second allele says. So if you're expressing no pigment, you will have a yellow lab. However, if you're having some pigment concentration being coded for, you will have a lab that just has a little darker of a face, a yellow lab that has a little darker of a face. Now, if we are coding for pigment, then it's really going to matter what the B's are saying. So anytime you have a capital B, you're going to get a black lab, and anytime you have lowercase b's or less pigment, you're going to get a chocolate brown lab. Polygenic inheritance is when you have multiple genes, so more than one gene that is determining the phenotype of human traits. And so a lot of human traits are going to be inherited this way. Hair color, eye color, skin color, height, weight, we think maybe intelligence. But in this case, you would know that something is polygenic when you see a lot of different phenotypes. And so if I took all of you and I, put you, on a continuum based on the skin color that you have in class, probably there wouldn't be two of us that have the exact same skin color. And that's because there's so many genes that are playing a role into what that skin color is. And so it's kind of like a dosage effect based on what type of melanin you have, if you have eumelanin or phomelanin. And so here we can see that, two parents that are heterozygous, this is like a really simplified way of looking at it, but, they could have a child that would inherit all recessive genes and not be coding for a lot of melanin and would have lighter skin, or they could have an offspring that would inherit dominant traits for all those genes, coding for more pigment or more of that eumelanin. And so this is how we kind of see it turning into like a histogram type of plot, where you're going to have more people that are somewhere in the middle, less that are on the extremes. And obviously, that has to do with, what part of the population you're in and where those people are located geographically. But this is just showing you kind of an example of how we determine skin color or height or weight. Now, albinism is the only exception to this rule. It can be inherited as a single gene trait. And so what happens is you're coding for this melanin or that brown color. And so what individuals who have albinism are doing, is they are blocking the production, or they're blocking an enzyme that would normally take this tyrosine to code for melanin. And so because they don't have that enzyme, they are unable to take that tyrosine and produce melanin from it. So sex-linked traits are another type of, different inheritance pattern that we see. And so when we talk about sex-linked traits, we're talking about genes that are located on the sex chromosomes and not, so those that are on the X and the Y and not ones that are on the autosomal 1 through 22. This is something that was discovered by T.H. Morgan using fruit flies. And so we'll kind of talk about exactly how sex-linked inheritance works. So just a little review that autosomal chromosomes are 1 through 22, and then your sex chromosomes are that 23rd pair down there. Now, if two humans come together, we know, that a male has an X and a Y, a female has an X and an X, and if you put that on a Punnett square, you would have a 50% chance of having a female and a 50% chance of having a male. Now, females are XX, there is some gene redundancy there, and so we're going to talk about kind of like how females silence one of their X chromosomes so that they're not expressing too much of those proteins that are found on those chromosomes. And then the X and the Y chromosome really has no redundancy. And the reason why is because the Y chromosome is really kind of just a sex determining region. It turns on genes for productions of male hormones. And so the Y chromosome is really going to code for a lot of those hormones, and there's not going to be a lot of other genes that are carried on the Y chromosome. However, the X chromosome, just a really small portion of it is going to code for kind of that sex determining hormones or products that you see in females. But we are going to see a lot of other genetic traits that are on the X chromosome. And when we map this, interestingly enough, there are a lot of diseases that are traced to the X chromosome. And so when we say something is sex-linked, we usually mean it's X-linked. There isn't going to be a lot of Y-linked diseases or just even Y-linked traits, but we will see a lot on the X chromosome. And so this is a colorblind test. So a person who was colorblind would not be able to see the numbers that are present in those circles there. But it's a good example of something that's sex-linked in humans. And so if you have a female with normal vision, but is a carrier for colorblindness, and she marries a male with normal vision, and if we set up that genetic cross, we would get 2 normal vision females. They have capital N's. We would get 1 normal vision male, and then we would get 1 colorblind male. And so here you can see that a lot of times sex-linked traits are going to affect males more than they affect females. And this is just for the plain fact that they only have one X chromosome and females have two. And so females can be carriers and males cannot. We see probably the most famous sex linkage family is the royal family in the countries of Europe. And so Queen Victoria, she was a carrier for hemophilia. And because there is so much inbreeding in royal families, as the generations went on, they started to see more and more people affected with hemophilia. And so, you can kind of see there how many males in that lower bloodline are affected with hemophilia. And that's because it's a sex-linked trait. It's carried on the X chromosome. And so if you would have a normal male, like Queen Victoria's husband, and her, a carrier, when they have children, every male that they have is going to have a 25% chance of being a hemophiliac. And so that just kind of shows you again how it can, it will just come up more in males and families than females. And so those aren't the only traits that will show up in males more. Male pattern baldness is another trait that is carried on the X chromosome. And so, for a female to be bald, she would have to carry 2 recessive traits, but for a male, he would only need 1. And so that is another sex influence trait that we see. X inactivation is the way that females are able to silence one of their X chromosomes so that they're not seeing too much expression of proteins from having that double dosage of the X. And so one X is going to become inactivated during embryonic development. And this is random. We don't know how one X is picked over the other, but whichever X is going to be unused or condensed, we call that a bar body. And so you can see here how this cell, you have a bunch of these bar bodies or condensed chromosomes happening. And it's random. We don't really know, which is going to become that bar body. And so this turns a lot of traits into these mosaic traits where you have some cells that are expressing one color and some that are expressing another, and we see this most often in calico cats. So the orange and the black are carried on X chromosomes. So in males, you can only have black or orange cats, but females, because they silence, because they could have both a black and an orange X, and you never know which one is going to be silenced, they will have kind of that patchwork quality on their fur. And the last thing we really need to talk about today is just environmental effects. And so there are some genes that can be controlled by environmental influences. One that's a really great example, you might have these, at your house at home, but hydrangeas, based on the amount of nitrogen that is present in the soil, that can change the expression of their flower color. Arctic foxes are also interesting. They will have different gene expression based on the air temperature. And so when the air temperature is really cold, their genes will express a white fur color. But when it warms up in the summer, they are going to express more of that kind of brownish coat color so that they can blend in with their surroundings better. And then the last example is really in humans, and that would be with skin color. And so we know that, in the summer, the more time that you spend in the sun, the more pigment you're going to have in your skin. And that's because those UV rays activate the production of melanin. And so, you're going to have more of that brown color in your skin from more UV exposure. And so that's all we are going to talk about for our advanced genetics, you have a problem set over these types of questions. That's called genetics problem set 2. That will be due next class.